Science Tonight audience. I'm not Chris Smith. It's not him. My name's Adrian Smith, no relation, but Chris is out this week on vacation, so I am taking the reins tonight. I'm coming to you live from my lab, actually, at the museums, my office at the North Carolina Museum of Natural Sciences. You might know me from there. You might know me from YouTube, on my YouTube channel, Ant Lab, or you might not know me at all. So if you don't know me, hi, my name's Adrian. I work at the museum. I'm an ant scientist. I study ants and other insects. And tonight we have an awesome program for you with Ian Grace, who is a marine ecologist, a marine biologist, and studies deep-sea larval dispersal is what he's going to talk about today. The good news is it's going to be an awesome talk. The bad news is I don't know anything about this subject. So we're going to have an awesome conversation, and I'm going to learn a lot. I'm sure we'll all learn a lot about what it means to study deep-sea larvae and the whys and the hows and what he's interested in and the cool science that he's doing. So one of the ways that Ian does his research is on this famous submarine called Alvin, which is something I have heard about, despite knowing almost nothing about marine biology. So I thought Chris and I thought it would be a fun way to start the program today to ask questions about Alvin, but not that Alvin. Let's ask questions. Let's do a little quiz about Alvin and the chipmunks. I mean, why not, right? We're just having fun. Okay, so Ian.
Yeah.
How do you feel about Alvin and the chipmunks?
I'm not there with you. No,
fantastic.
That's the hot seat for me.
Okay, let's go. Let's just jump on it.
Sure.
And tonight, if you're watching on YouTube, you can enter questions in the chat, talk to us there. I will bring your questions right to Ian. Look, I'll write something in the chat now. It'll come up as Ant Lab probably. Let's see. Hi there, it's me. Enter. Bam, I'm in the chat. So I'll be watching the chat all night. Let's start with this quiz. Are you ready, Ian?
I'm ready, yeah.
All right, number one, here we go. Who are the chipmunks Alvin, Simon, and Theodore named after? Multiple choice here. The creator's real-life sons.
All right.
The names were chosen at random from a phone book, or the executives of their original record label. What do you think?
I think I have to play a game here. Which correct choice would you place where? Are you an A, a B, or a C person?
It's even harder because I didn't write these questions. Chris did. So
great.
Yeah.
I'm gonna go with the record labels. Yeah.
Oh, good. Yeah, that's right.
That
that's right. Yeah.
Right, okay.
Alvin Bennett, Simon Waronker, and Theodore Keep were the chief executives of Liberty Records at the time.
There you go.
I know.
Okay. I typically have bad luck with these sorts of things.
Well, you're 1 for 1. You're killing it.
Right. I should have put C's on all my tests through high school and college.
There you go.
I would have got
okay. Number two.
Sure.
Let's see. Which of the following songs was never covered by the Chipmunks?
Oh boy. All right.
Famous recording artists on Liberty Records, as we just figured out.
Yeah.
All right. Option 1. Never recorded. Achy Breaky Heart by Miley's dad, Billy Ray Cyrus. Beat It by Michael Jackson. Walk This Way by Aerosmith. Which one wasn't covered?
Wasn't covered by the original
by the
people
Chipmunks.
Or by the Chipmunks? By the Chipmunks. Yeah. In any movie, so we can see the song could have been covered in the movie, and that counts as being covered.
Yeah.
Okay. So I feel like Beat It and Walk This Way would probably make their way into a movie. No,
okay.
Unless they're really
so
hard.
You're going with Achy Breaky Heart?
Yeah, I'll go with that.
Oh man, you're wrong. Sorry.
It must have been that the song was tough to get with copyright or whatever.
Maybe it was Walk This Way, Aerosmith. Chipmunks didn't cover the Aerosmith song.
Okay.
Man, yeah.
I would imagine that being in a movie somewhere. That's a shame.
I know. All right, let's do one more. Ready?
Okay. Yeah.
If you turn on the radio now, Christmas music has already started infecting our airways.
Yeah.
So, Christmas theme. The Alvin and Chipmunks are a Christmas staple in Christmas music. So, according to their song, the Chipmunk song, 1958, what does Alvin want for Christmas? Peace on Earth, a hula hoop, a harmonica?
He wants a hula hoop.
Oh,
Yeah.
you got it.
Yeah,
Excellent.
I've got that pitch that he accesses there in that upper vocal range, just
Great.
living in my mind over Christmas, sadly.
Yeah, the fun fact there, I guess, is that song 1958, and the hula hoop was
Wow.
invented and introduced to the public in 1958.
Really?
It was a fad. Yeah.
I did. Wow.
The
That's
hula
a
hoop.
lot older or younger than I expected to be.
I know. It's in the long lineage of fad Christmas presents. Tickle Me Elmo and then hula hoop way before it, right there.
Really?
Yeah.
I would never have guessed that. You should have asked that as a question. Which came first?
Tickle Me Elmo or hula hoop?
Yeah.
Okay. Next time. All right, we've gotten way off course. That's not even science.
I don't study hula hoops.
We're adrift in the sea. Okay, so let's bring it back to deep sea biology, and you're going to talk about deep sea dispersal and deep sea larvae, right?
Yeah, that's right.
Okay, why don't we start and give me your sort of elevator pitch and what you're all about. We'll just jump into it.
Sure. Yeah. So basically, what I study on the surface level, as a pun there, I essentially study the movement of animals in the ocean, and the animals that I study are mussels that live at cold seeps in the Gulf of Mexico and what we call the Western Atlantic margin, essentially the U.S. East Coast. I'll share a picture with you. Okay, so you should be able to see that there. And the top left, you can see cold seeps are found at passive margins, at active margins, along continental margins. That's generally where they're found. And the top right there, you can see that bubbles from the seafloor are coming up there. That's actually sulfide and methane that have been converted from hydrocarbons by subsea floor bacteria to be emitted and to be utilized by the communities that are found at cold seeps. So actually the carbonate that's emitted there actually precipitates out of the seawater to form that rocky seafloor. And then the mussels in the bottom right there, and actually just to make a pit stop, the bottom left is a gigantic bacterial mat, which you also find at seeps. But the bottom right, the mussels there actually have bacteria, symbiotic bacteria in their gills that consume the methane and the sulfide that's emitted from the seep and allows them to survive, as well as along with their typical filter feeding behavior. So what I study is the dispersal of these mussels' larvae, and that's a natural mechanism, larval dispersal, that drives their species distribution and their population persistence. So larval dispersal refers to the movement of young from communities, populations, adult populations. It refers to the movement of the young to basically just different sites or to the same site that they were born at. And so that's referred to as natal retainment or self-recruitment for a site. So that happens as well as larvae dispersing long distances here. So what I do is I study larval dispersal by looking at shell geochemistry, and that geochemistry is of the shell, and the shell serves as a record of environmental conditions that the shell experiences throughout growth. I'll get into why in a little bit. But larval dispersal is really important because the degree to which dispersal occurs determines how isolated or connected a given population is or could be. So the result of larval dispersal is called population connectivity, and that can help measure how resilient, for example, a population is to disturbance, whether that be anthropogenic disturbance like mining or natural disturbances. So this type of work, larval dispersal and connectivity research, have a lot of conservation and resource management implications, both in terrestrial environments with just connectivity, but also in the ocean and the deep sea in particular, as it's not so well understood how we should conserve the deep sea, since it's a fairly new environment for us to learn about. Yeah. And for those of you who might be familiar with connectivity, but only in the terrestrial sense, essentially it's the same idea. You have animals moving from one place to the other. And in terrestrial ecosystems, we talk a lot about landscape connectivity. In the ocean, larval dispersal is sort of the equivalent. You can still have landscape barriers, but larval dispersal is the mechanism of connectivity in the ocean. And so that's why we're studying it as a precursor to understanding connectivity. So
Ian, is this map?
Yeah.
Are these where you're doing your work at? Are these the deep sea points, like south of Louisiana and the Gulf, and then on the Atlantic side of the East Coast?
Yeah, so I'll show a site map later, but in general, yeah. So the Gulf of Mexico here on the left, yes, and south of Louisiana on that continental margin that you can see. We don't have so many sites that are out in the abyssal zone there. Most of these sites are on the continental margin with canyons that are incised or right next to them or on the very top of them. And then we have some around the Florida escarpment as well and on the East Coast there. But yeah, generally it's all along the continental margins. And again, I'll show a site map later with all the different sites. And we have 6 primary sites. Yep.
Cool. So in this map, the light blue versus the dark blue, that's the continental, what did you call it?
That's right. Yeah, the continental margin. So the light blue refers to that's the continental slope that you see, gently sloping downwards. And then there's a cliff edge essentially that just drops off into what we call the abyssal zone of the ocean. That's the majority of the ocean. If you look at a blown out view of the Earth or a given ocean, you'll see that this light blue area is just a thin little line, and then it drops off into dark blue or black. So yeah, the continental margin is just the edge there, and that's where a lot of deep sea canyons can be observed, and you can find a lot of interesting environments in those. Yep.
Yeah, cool. So tell us about your study region and the environments there.
Yeah. So actually, this study region is not really geologically active. It's a passive zone instead of an active continental margin, which means there's no tectonic activity really occurring compared to somewhere like the Pacific, where you have all sorts of volcanoes and plates moving around. That's where you'll find hydrothermal vents, for example. Many people have heard of those. Not so many have heard of cold seeps. Cold seeps form everywhere, but also at passive zones where you basically just have gas leaking out of the ground and then being utilized by animals that have managed to use the stuff coming out. And there are complicated reasons as to why those gases emerge, but this region is essentially passive. And I'll talk about some of the distances we're dealing with here as well. So if a larvae, for example, was to disperse from point A to point B, it'd have to travel in a straight line, and it's not a straight line in reality, but 400 miles if it was a straight line. And then if it wanted to get all the way around the Florida escarpment there and get up to point C, it'd have to travel about 1,200 miles. And if it wanted to get north to the northern sites that we have, I'll show you those in a minute, it'd have to travel on the order of 16, 17, 1,800 miles. So quite a distance. And a normal larvae just can't expect to swim that. So we know that Bathymodiolus larvae, Bathymodiolus being the genus of mussel, we know their larvae can survive in the water column for a crazy amount of time, 5 to 17 months. But in order to disperse these long distances, they have to reach surface currents. Some of them have to reach surface currents that allow them to disperse more quickly than they can swim. So what I'm trying to do is figure out why some species are in the Gulf of Mexico, but not in the Western Atlantic margin, or in both regions, but only at certain depths, and a combination of those. So for example, one species is found in the shallow to intermediate depth range, 400 to 2,300 meters in both regions, both the Gulf of Mexico on the left and the Western Atlantic margin on the right and towards the north of the screen. Another species inhabits both regions, but has only been observed at deeper depths, 2,200 meters to about 3,300 meters. We don't know why. And then again, a third species is found only in the intermediate depths in the Gulf of Mexico. And so we don't really know what's driving that distribution. We have a general quantitative sense of the connectivity. We can say that, oh, these sites are not so connected to these sites, but we don't know why. And we need to understand that. Otherwise, we're never going to fully understand connectivity to a high resolution. So one hypothesis that we have to explain this pattern is that different species will disperse different distances depending on the depth at which they disperse. So depth of dispersal is a phrase we toss about a lot in this research. So that is to say, depth of dispersal matters. Mussels at greater depths, for example, might retain their larvae at their natal site more so than those in shallower areas that could pretty easily reach, those surface currents that let them go way far out there. We're not sure, but that's what we're trying to figure out. So I mentioned I use a geochemical approach to study larval dispersal. There are other ways, but studying larvae directly is very difficult. So I don't look at the physiology. I don't look at behavior. I more or less infer dispersal and these complicated patterns that result from things we don't understand. I use a geochemical approach to look back on what's already happened.
That's cool.
Thanks.
So geochemical approach, that means you're studying the chemicals in the geography of the ocean relative to the mussels themselves? Is it kind of like studying like I don't know, like almost like digging up a fossil and then kind of studying the rock matrix around it to infer stuff about it.
Yeah, that's a good way to describe it, I guess. So in each of these places that we've collected larval shells from, there is a chemical profile in the shell, but also the water column. What we're trying to figure out is more or less where these larvae have come from based on their composition. So we're not analyzing the water chemistry so much as we are the result of that water chemistry, the water chemistry that's been applied to the shell through its growth. And I'll talk about that in a minute and why that happens. But essentially, as a shell migrates from, for example, point A to point B, it might move through different chemical gradients, maybe salinity and temperature change over the course of its migration. Those things control, to some extent, the elemental uptake, or let's think of it this way. Those things control the metabolism and the behavior of the mussel, and those factors will change the elemental uptake, how it grows. You could almost kind of make some social comparisons. You know, if you grow up in certain places, maybe you're going to think these things or do these things or be, this way, whereas another mussel might be different. And so we're basically trying to compare that and see if the composition can tell us anything about where these mussels are going.
Can you tell, like, does a mussel grow like rings? Like, is it like tree rings? Like, as it gets older and older, deposit as extra things, and you can tell, like.
So actually, this is the life cycle. I'll get into that in just a sec. But so through development, the shell forms a record of the salinity and temperature. Just for example, those are two variables, and then we can use those that chemical composition resulting from those factors to kind of infer what environments the shell experienced. So gametes are released. This is the mussel life cycle. Adult mussels will release gametes, broadcast spawning into the ocean, into the water column. And then there are a few stages, but eventually we get to this first stage called PD1, which means prodisiconch 1, a long, complicated word that essentially just means the start of the shell. And it's that little yellow circle there. We're not referring to the entire shell in that picture, just that little circle. And it's white, it's extremely small. That little part is probably about 50 to 70 micrometers or less. And much less when it first starts growing. So it's accumulating elements in this stage already, and it's dispersing as well. So as soon as the gametes are released and they're fertilized and you're starting to form the shell is moving through the water column somewhere, whether it's staying at its natal site or traversing somewhere else, it's moving. Then it forms this PD2 shell, which is a larger shell essentially, but also accumulates elements that we're interested in looking at. And then finally, that last one, that disiconch in the very bottom, almost looks like a fingernail attached to the PD1 and PD2 shell. That forms when the cell has already settled on the bottom, when it's attached to another mussel or a rock or something, and it forms that settlement shell. So in all of these stages, we can target it for geochemical analysis and say, oh, well, when it was growing the PD1, can we figure out maybe where it was or what sort of environments it was experiencing at that stage? And same for PD2. And the disiconch, can we figure out at that particular site, what does the geochemical profile mean for the composition of a shell? And then also, if we analyze the adult shell, way after the larval shell and the juvenile shell, if we analyze the adult shell over time, we can figure out, hey, does the chemical profile at each site change over time? does that mean anything for, if it does that mean anything for the composition of the shell over time? It helps us to refine our data a little bit. Mm-hmm.
Yeah, cool. So what would make a different like chemical environment? Like is it like sea levels? Is it the rest of the marine ecosystem? Is it what would change the chemistry of the environment?
Yeah, virtually everything. I'm not a chemist myself, nor a physical oceanographer, but I can say that, yeah, the chemistry would change as a result of physical currents changing in the region. Canyons, for example, will have this, it's not the correct terminology, but inflow and outflow through the canyon, the water changing. Methane seeps themselves are obviously, they have a lot of chemicals going on there. And so, yeah, they can change and die over time. I'm not sure if that would affect composition or not. I would imagine to some extent. The primary factors that have been researched as changing composition, as having some significant effect on composition, have been salinity and temperature.
Cool.
So
by looking at the chemical composition of the larvae, you can tell like where it originated from or how far it's been traveling or dispersing.
Mm-hmm. Yeah, let me see if I have a slide that refers to that. Yeah, so yeah, in shallow environments, that's been really popular. That's been a popular approach. So a shallow environment so you can go into like an estuary, for example, and pick up some larvae and analyze their composition, and they're easy to access and easy to collect. And so you generally know where those larvae developed. And then you can go somewhere else and pick up other larvae and see, what their composition is. And you can compare the two and you can figure out, this shell, you can form what's called an atlas, a geochemical atlas, where almost any given place that you choose on a map in an estuary, for example, you can say, I know what the composition of this particular species shell would look like at this particular point in time. And you can compare that and say, I see a signature that looks like that, but it's over here. So maybe that shell came from over here, and maybe we can draw that line there and figure out that that's a dispersal path, for example. In the deep sea, you can't really do that. There are a lot of limitations to studying anything in the deep sea.
Yeah, so you're gonna show us some slides and pictures of like how you actually get out to sea and what it's like to actually study this stuff.
Yeah, that's some preliminary results. So yeah, this is RV Atlantis here, an extraordinary ship. It was actually the namesake of Atlantis, the space shuttle. And there was a sailboat version of this a long while ago. That's her from the back. This A-frame actually, the gigantic two towers in the back, that actually holds the sub and retrieves it and deploys it. And then there's tracks on the actual ship that go into a hangar, and the sub will roll into the hangar. Okay, so these lines indicate kind of where we stayed. So the very bottom there, those were some quarters for us that we didn't really stay in very long. The red line is basically where we did all of our work. That includes the labs, the computer science rooms, the prep rooms for Alvin, and so forth. And then the other two lines are the mess hall and sleeping quarters. So these two areas are basically where we spent 99% of our time. So the left is the main lab, just a long, almost ship-long lab with a bunch of tables and storage places. And then the right here is actually just in front of the sub. This is what we called the bio box, which it held all the animals and specimens that we collected over the course of a dive. You'd just pick them up with a manipulator, the arm, and drop them into the box and close the box. And then it was our job to pick up the stuff from the box and take it back and sort it and do all sorts of things with it.
That's in the front of Alvin?
That's in the front of Alvin, yeah. So I've got a picture somewhere here. We'll get to a picture that shows that that's in front of Alvin. Mm-hmm.
Cool.
In the top left here, you can see us sorting. These poor people would stay in a room and just sort. They're looking through dirt to find mud and dirt to find larvae, so we can take them. Larvae, Bathmodiolus larvae are really low density and very small in the water column. So you know that needle in a haystack dilemma?
Yeah.
It's like that, but it's like a needle in 8 haystacks trying to find one of these things. So it's very difficult. We go down there and we basically vacuum in between the muscles, the interstices of muscles, trying to find these things. The best we can do is get a pile of dirt and water and try to sift through it and sort through it and look for larvae. Best we can do.
So we have a question in the chat from Fred,
Cool.
who asked basically just that. Like, you're studying larvae, and he asked, how do you collect them and do a chemical analysis in such a small organism? And it sounds like, tell me more about the larval sucking vacuum. That's how you're collecting. That's how you're collecting them.
It's one way, yeah. So that's one way. I should have included a picture of the vacuum, but that's one way we actually do it. It is basically a water vacuum where we're just sucking. It's a little tube, and we just poke it down in between the muscles, and we suck up a bunch of stuff, and you can see all kinds of material coming up, and it collects it in a jug on the sub, outside it in the basket in the front of Alvin, and we bring it up. So we also
are larval muscles attracted to adult muscles? How do they get back to the
so
adults?
They'll settle on them. There's a lot of different cues that a larvae will follow to settle. A lot of that's unclear, and I'm not an expert in that to any extent, but yes, they'll settle on muscles, and so you'll have muscles growing on other muscles, and muscles growing on the carbonate outcroppings as well.
Wow, that's cool.
Yeah. So we'll also use a scoop to scoop the adult mussels into a bucket, and that's where we the bio box here, that's where we can collect those from. And we can collect other critters as well if we're interested in studying them. So some people were interested in sponge. And at one point we had a fish that actually managed to get in there as well and had a pretty bad ride up. But yeah, a lot of things get in those boxes. There's some other tasks we do, like sieving through things. And also we made video highlight reels every day, which helped us kind of get a qualitative idea of how the day went and how the dive went. So here's us sorting some. This is essentially just a slideshow that I'll comment on. We're sorting some mussels here. We're measuring them and taking some tissue for analysis. Building equipment that we're deploying on the seafloor. Had to deal with a whole lot of weather, especially after we came around Florida onto the East Coast. We dealt with, it wasn't quite a storm, but it was extremely windy, and we got about 20-foot waves at a few points. So interesting living conditions then. We can play ping pong sometimes, especially if we have nothing to do, which was extremely rare. Otherwise,
wait. Ping pong on a boat.
Oh yeah.
Ping pong on a boat seems extra challenging.
Extra challenging. You might not be able to see it. There's a little string there that shows kind of the angle we're hitting. I don't know what angle we hit, but it was enough to kind of, we would be walking out of the mess hall sometimes, the dining room, and typically walking and then just, we're sliding down the dining hall and trying to not hit the person as we come around the corner. Wow. Yeah, ping pong on a ship. It's possible. Makes for very interesting curves and turns with the ball.
Yeah. So who's on the ship? How many people are on this research vessel, and who are they?
Yeah, so I think the ship can hold 36 science crew, I think. I'm not sure if that includes the ship crew or not, but there were 20 of us at this time, 20 scientists aboard, be they undergrad assistants or graduate students or postdocs or PIs, principal investigators of the project. Mm-hmm.
Cool. And there's the guy with the camera in these pictures.
There's the guy with the camera. So this was unusual. We had a person that came and did a documentary with us. He was doing it for some sort of arts symposium, I think, in New York. And he had some ties to Woods Hole, and I don't think, it was just Woods Hole, I think. We weren't quite sure why he was there sometimes, but he was a very nice fellow and had a lot of stories to tell. But yeah, he had a drone and he had a camera, and we've never seen any videos or pictures of anything he took, unfortunately. I would have loved to. Yeah.
One day. When were these pictures? Like a couple of years ago, last year?
No, just last year.
Last year?
March.
Okay.
February and March.
Nice.
Yeah. So another thing we can do is we have styrofoam cups. We can drop them down to the bottom. So, typical standard styrofoam cup, put it down on the bottom, and it turns into, the height there is roughly about half of a pencil for a little bit of scale. And
Those
same way.
aren't ordinary cups. I see a cookout cup in there.
That's right. There's a cookout cup in there. There's also, if you look in the bottom left, there's a head. There's a styrofoam head. Those start out huge, and then when you bring them back, they're about very small.
Oh, I get it now. So this is like in this. You're not like putting this. You're not putting trash in the ocean.
Oh no. We have it in a bag and we bring it up. Oh yeah, no, I don't want to imply that.
Okay.
We do see trash down there, but no, we bring everything we use down there.
And this is from the pressure differences being in the sub.
Yeah, pressure differences. Yeah, the pressure you experience at be it 400 meters or 3,000 meters. I think the farthest we went was close to 3,300 or so.
Wow.
For those of you who use feet, that's like that's I think over 10,000 feet. My dive was I think it was like 2,600. I think roughly, maybe 2,300, over 7,000 feet.
Wow.
And yeah, the pressure you experience at that depth is nuts. It's really crazy, and it compresses these cups in this way. And you have to put paper balls inside of them because if you allow them to compress just free will, they'll just crumple into a ball. You have to have something that presses against the sides a little bit.
Do you still have your cup? Where's your cup?
Yeah, I do. I don't have it with me, actually. I left it at state. We have a whole bunch of them in the lab, just dotting the countertops.
Wow, what a cool tradition.
Yeah, I'm bringing them home for gifts for people and stuff.
Nice.
Yeah.
It's like the opposite of a moon rock.
It's
a deep sea cookout cup.
Yeah, exactly. The farthest away from that as possible. Yeah. This is the bridge. So basically the crew would let us walk around pretty much anywhere we wanted to, which was a very weird experience. This is where we slept. I was in the top bunk. I don't know how my bunk isn't made. I don't know why that happened. Bathroom to the left goes straight into the other room. So you've got two doors to close. And our wardrobe was to the right there. And we had a little tiny window to look out of in the middle there. Not very large. And this was for about 3 weeks. It took everybody a couple days to get used to not, don't wake up too fast because you're going to hit your head on the ceiling there. And everybody's going to hear it. This big, gigantic tin bang. And yeah, this is Alvin. It's a beautiful machine. One of the only pieces of equipment that is actually named. I don't think they were originally planning on naming it, but this was way before this version of Alvin, but somebody decided, hey, let's name it Alvin, and they did. But anyways, the center sphere there that you can see that's a titanium sphere, the silver coloring there, that's the titanium sphere. And that's what we're in when we're going down in the sub. And those blue circles, those panels are actually covering windows. So we don't want to damage them. We don't want them to be inflicted at all. But those cover the windows, and those are the only points, except for obviously the hatch, that are not fully sealed behind this thick titanium sphere. And this thing is engineered to an extraordinary degree to be safe. Everything else that's attached to the sphere is stuff like syntactic foam, arms, the basket, basically everything else, and the paneling that covers the foam. All that can be detached. I think I have another good picture here. All of this stuff can be detached, not just by taking it off manually, but through computers on the sub. You can eject it. So you could return in that sphere if you had to, for example, in a safety situation, an emergency.
Are those windows exposed? Can we see like in the middle of the titanium? That's what the window looks like?
Mm-hmm. That's what the windows looks like, yeah. And so when you're inside for scale, you're covering your eyes like this, and you're looking into the window like that because you don't want your eyes or your glasses, for example, to touch the window itself because they don't want it to scratch. So you're huddling up to the window like this, and they're very small. Maybe your hand would probably cover it if you were to put it up against the window. It's very difficult to look out of it.
When you're like collecting the larvae off the beds, you're not like visually navigating. Like how do you decide where to move the collecting? equipment?
That's a good question. Yeah. So when we're down there, one, we have a set list of objectives we're trying to accomplish. So the person who's sitting left of the pilot, and the pilot sits directly in the middle and the front. So he sits right in the middle there. He looks out of that front window, and he's scrunched up. Best I can kind of imitate. He's scrunched up with his knees up here. He's driving the sub like this, and he's looking out the window. It must be extremely uncomfortable. The two observers are sitting next to him, kind of reclining a little bit and doing this kind of turning motion to look out the windows in both directions while he's maneuvering around. So we have a decent field of view, especially if we keep our eyes glued to the windows at all times. But the pilot is always looking out the window, and sometimes they'll look over to a different window just to make sure we're safe and we're going where we need to go. And so when I went down, I was telling the pilot, we need to do this, X, Y, and Z. And can we go and tackle that objective? Can we go over here and collect that particular mussel or that particular organism? So we have an idea of what we want. And when we're down there, it's always a little bit surprising. Just trying to do the best we can. Hey, let's go suck that up. It's stressful and it's intense, but there's also this kind of funny casualness to it because we're down there in an environment that is very not poorly explored, but underexplored. And it's just, hey, let's take advantage of the situation. Let's go and take that there. Let's go and pick these mussels up. Oh, actually, can we go over there? Yeah, it's interesting. But that's how the pilot navigates. He's just craning out the window like that, and we're trying to help him by looking out the side windows.
Cool.
Also, I forgot to mention here in this picture here, you can see all these cameras and lights, and we utilize all of them. So there's cameras we can control from inside the sub. That's actually one of the jobs that we have as observer. And we can pan them all around and zoom and everything. And that's how we take a lot of the video. And we also have camera mounted on the basket in the front. There's cameras everywhere. So we can sort of navigate from those as well. Look at certain things, point at certain things. There's laser pointers as well. Yeah.
Nice. Let's it's almost 7:40. So
Yep.
you have a video that we want to get to.
Yeah, actually let's do that.
Yeah. And then we'll go more into, we can revisit some of these slides and then do question and answer too.
That'll be a good way to answer some of the questions revolving around Alvin. Yep. So this dive video, I'm going to narrate this. There's no sound. I'm going to play it in a few seconds. But this is basically the idea of a dive, and maybe the most interesting thing I show all night. If you take nothing else away, at least take away the fascination of this. So I'll start in 3, 2, 1. So this is how Alvin is deployed. It's lowered down on this rope, this tether, and then they remove that tether off of a cleat. And they're looking inside the sub. The hatch is closed. They're picking up a phone. It's a wooden phone, so it doesn't have anything to do with, it doesn't mess up with the water. They're talking to the pilot. Eventually they get off and we're down and we sink down to the bottom. And here we are. There's a brine pool to the left there, salt. We have a lot of this going on where fish come up and bump the lens or bump the sub. And that was actually an eel. Those are some mussels. And the little white bits you see there are actually snails. I don't have to tell you what that is. We saw a few of those. And they're just absolutely beautiful and tolerated us really well. These are hagfish, a little bit creepy, I think, but they're currently swarming around a fish head. Hagfish are extremely slimy and they'll knot their bodies up and little creatures, but they're amazing. So here's how we scooped up mussels. We just had a little basket and we scoop them up. And then we just drop them into a box right there. And everything down there happens quite slowly. It feels like it's going quickly when you're there, but it's quite slowly and it's quite predictable and intentional. So here are some tube worms, another sort of dominant community you can see at seeps instead of mussels. There's a crab on top of the tube worms. I wish I could tell you about tube worms. They're really fascinating. Here's a little hagfish, curious little hagfish. He smells some food in the bio box there, not for him, but he thinks it is. Here's some little galethid crabs jumping around. Kind of looks like something out of a Disney movie. Not quite sure what this is. I think it's some kind of tunicate. I'm not sure. It kind of looks like a salp. The reason we videotaped it is because we don't know what it is. This is beautiful. I think the most alien thing I saw down there, not in first person, but through video. Opheroids, just hanging around on some... And then we get out. We walk down some stairs and we're done. And that's a dive day, essentially.
Wow, that's pretty cool. What a science adventure.
Yeah, it is fascinating. It's very different than anything I've experienced before.
Yeah, that's really cool. Okay, so I want to get to some of the questions that popped up in the chat.
Yeah.
Lisa is interested in the effects of sea level rise and maybe climate change and how that might be impacting these populations or even the dispersal of what you're looking at.
I have to preface with I'm not sure. I don't think sea level rise itself would directly affect dispersal. To my knowledge, a physical oceanographer could probably provide more detail on that. But I don't think, because these are deep sea environments and are actually somewhat isolated from what's going on, we do still have acidification going on. I'm not sure if temperature really changes so much in the deep sea. I think it's a more stable environment. Possibly in terms of, I'm just kind of guessing here, but larval behavior, if you have larvae that are closer to the surface and are experiencing more fluctuations in temperature due to climate change or other issues, or possibly salinity, yeah, you could potentially probably have some alteration of dispersal. That's my best guess. I'm not sure if that's applicable to Bathmodiolus larvae, which, while we say at the surface, aren't way up there and at the surface layer, they're pretty deep still. I'm not sure how climate change would really affect that, or at least to what degree it would. But it's probably safe to say that somewhere along the line it does. One thing I think that's interesting is those cues for settlement that I talked about earlier, whether they sense temperature differences or maybe salinity differences or something else to settle at a site. Those could probably be affected by climate change somewhere along the line. That's my best guess.
Cool, thanks.
Yep.
All right, Fred wants to know about the chemical analysis from such a small organism. Yeah. I'm curious about that too. Like, do you have to, like, when you're pulling these things up from the bottom of the ocean, do you have to bring them on board and do the chemical analysis right away, or freeze them?
No, fortunately. So what we do, just to give you the process of what we do, we take them, and the adults will distribute them among people, and we'll take out the tissue and so forth. And then I just have a shell. That's all I have, and it's fairly dry. And I'll wrap it up in some foam, and I'll put it into a bag with other ones. So they're not bumping around and touching each other and rubbing off onto each other or anything, or breaking. Pretty fragile and thin. That's the adult shells. They're bagged and they're frozen. You can put them in ethanol, but some of these shells get huge, so you don't want to do that. You want to freeze them. So that's what I do. Freeze them, just standard freezer, negative 20 Celsius. The larvae can go in ethanol, but I just freeze them as well. So they go in little vials. Once the very dedicated folks have actually sorted them and found them, they just go in little vials, and I put them in the freezer. Back at the lab, so at State in this case, I will go in and I will split the shells. And an assistant of mine will split the shells, so we have only one shell instead of a whole shell. We just have one shell. And then we'll mount that shell face down on a glass slide after we've cleaned it with a little bit of acid, some milky water. Then we'll take it to what we call an LAIC PMS facility. And this is essentially laser ablation plasma mass spectrometry, inductively coupled plasma mass spectrometry. For those of you who don't know what it is, I don't think I have the time to describe what it is, but take this away, that we're shooting a laser at the shell, and we're taking a very, very small portion of the shell and filtering it through an electromagnetic field or electrostatic field, which sorts the material we've injected into it, into its ionic components, and it can detect the curvature at which those ions curve through the machine, the detector, and it'll say, oh, well, I've seen this, this pattern, this curve pattern happen so many times, and so you have X, Y, Z, you have this number of this particular element. It's complicated, and I don't fully understand it, but that's essentially what's happening. We're measuring it with something called spectrometry. That'll tell us what concentration of a given element we have, and we're measuring all sorts of elements. We standardize that to calcium because that's abundant in the shell. So that's how we standardize it. And we try to have as many samples as we can.
Cool. Yeah. Nice. So the answer is lasers.
Blasting
the thing apart with lasers.
Yeah, it's not the only way. You can ingest it with acid and then inject it into the machine. Laser ablation is a really cool way, but it is just a way to process your sample. Mm-hmm.
Nice. Okay, so Troy's got some interesting questions here. Okay. Wants to know two questions. If DNA is ever involved, like have you ever taken DNA samples to compare populations?
Yep.
And then you showed a picture of the mussels and there were white spots on it. Just interested to know what those things were.
Yeah, was that the? I can share my screen again. Well, to answer the question about DNA first, so that's genetics. And genetics are fundamental to connectivity studies. And in fact, when we were proposing this, some of the reviewers said, well, we've already kind of done connectivity in this area, especially with genetics, and why should we study it in this way? Genetics are great for connectivity, but they don't really inform on larval dispersal because they are two different things. Connectivity is the result of larval dispersal. And to study larval dispersal, we can't really use genetics. We have to either use geochemistry or isotope approaches. Maybe genetics can come into play, but those are the approaches we're taking.
Right, because your questions are like concerned with like the life of a single mussel, right, basically, and what it experiences, right, not what's inherited, right, through population structure or whatever.
Exactly, and connectivity, like I said, has already been somewhat quantified for the area. We can generally say, well, this area is kind of not as connected as this other area, maybe, or at least these populations are not so connected. But to understand why that occurs is almost more important, and to resolve that connectivity to a higher degree. So for example, we're trying to make biophysical models. We can plug in the variables of larval dispersal that we think are the case, but we don't actually know, and that's physiology, behavior, and so forth. What I'm trying to do is kind of confirm what we see and also maybe provide some more inference on what's going on with these particular strategies. Yeah. So yeah, genetics are great, but not exactly what we're looking at right now.
Cool.
So about the mussels with the white spots.
Yeah, I think we got a couple of questions we can get to when we go back to the
okay,
pictures. Yeah, but
Cool.
let's start with the muscle spots. Yeah.
Sure. Maybe I can find an image of the mussels standalone, maybe. Was it in the beginning?
I think it was during the video.
During the video. Okay, I
Yeah.
can skip into the video somewhere.
There we go.
Here.
Perfect.
Yeah, so these are actually snails. And I don't know if the resolution's good enough, but they do look like snails from here. But yeah, they're just attached to the mussels and running around on them. I'm not entirely sure. I wish I could inform you more about the snails and what they're doing there. And I think what they're called are Bathynorida natacoidea. That's their scientific name. And we do study those as well. And you can actually do geochemistry on those shells as well. Same idea. You can do this sort of geochemistry on any calcium carbonate structure, including otoliths, vertebrae. So shark vertebrae, for example, has been one thing that's been studied. Not for the deep sea, but yeah, otoliths is a really common one.
Cool.
Snails,
So if there's snails, there's another question here that's kind of pertaining to that. If there's other organisms that are like living on the ones you're studying, do you think your questions are addressable by studying other deep sea things, or is it specific to mussels?
It's not specific to mussels. So yeah, actually, there are other species, other animals that disperse, other invertebrates that disperse. Trying to think of a couple. There are other mollusks, of course, the snails being one. There's a whole list of different.
Are there like deep sea corals that do it?
There are deep sea corals, but I don't know how they disperse. I don't know anything about deep sea coral, and we're not studying those in particular. If I pulled up a proposal, I'd be able to refresh my memory as to what exactly all the different animals are. But yes, there are. This is not just about mussels. This is about, in general, we can extrapolate our findings to different organisms, different animals. Yes.
Yeah,
that live specifically in the deep sea. Mm-hmm.
Another question has popped up about other studies being done with this stuff. I'm wondering if is there like a deep sea lab rat that everybody's studying? Is there a deep sea fruit fly that like all the cool kids are down there in Alvin studying, and then you're just over in the corner working on mussels?
Well, actually, that's funny you phrase it like that. Yeah. One of the mussel species, this particular region actually is really well studied compared to the rest of the deep sea. So it's kind of like a really good, foundation to study something related to the deep sea, is to look at it in this area. And then second, one of the species of mussels that I'm studying is sort of, if you had a lab rat, is basically that. It's called Bathymodiolus childresi, and it's really well studied. And yeah, kind of everything that comes along with something being fairly well understood, we can draw a lot of implications of results and conclusions of other papers, be it the physiology of the mussel, the connectivity of the mussel, life history, and so forth.
So the mussels are the lab. So you're like the deep sea fruit fly. So what's like the deep sea weird thing that almost nobody studies?
Ooh.
There's a suggestion, maybe tube worms. Are there no tube worm biologists?
There are, I think. There's a person called Craig Young, a researcher, Craig Young, who is from Oregon State, and he's an incredible researcher. He's the chief PI of this project, and he would be able to answer every single one of these questions. But he studies tube worms as well as mussels and snails and all of these other creatures. Tube worms are one of the other species that I mentioned that they do disperse, and we don't know anything, I think, about their spawning. Very little is known about their spawning. We were really happy to see, we actually saw spawning occur. We saw the little jet of gametes from tube worms actually occur, which is rare. And Craig was really interested to see that as well. So he studies tube worms. And yeah, that might be considered one of the more mysterious species. There are others, but I wouldn't even have names for them. There's a lot of things you see in the deep sea that make you just question, what is this? What is this doing? Why do we see this happening?
Yeah,
It's one of the
my jaw was on the floor when you showed that like coral, like that fan shaped thing with the
fan coral.
what was off of it? Can you pull that up again?
I can,
That just
yeah.
looks so
It's very
bizarre.
alien. Yeah. I do not know what they are exactly. I think they are either brittle
Wait,
stars.
so that's not all. So what's going on here? There's a
So that's
coral, and then
it's fan coral, yeah, which is fascinating
okay.
in the first place because it's deep sea coral, which is like, wow, you wouldn't expect coral to be in the deep sea. And when I first learned about that in undergrad, it blew my mind. But the other thing is, yeah, and I've never seen this before, is well, I think these are called Opheroids, Ophurodes, I think. And yeah, they're just grappled onto the fan coral. I have to admit, I don't know why. Feeding probably.
And they're moving, like you can see them move and
Yeah, they're moving.
Squirm
Yeah.
and wiggle and stuff.
Yeah.
Oh, that's whoa.
It's alien, isn't it? I share this more to just emphasize the kind of alienness the deep sea takes on sometimes. I wish I had a better understanding of this to share with you, but I don't. We see animals like this that have the little arms. They're one of my favorite. Those are Opheroids, and you can see them on the bottom a lot. And they're just little brittle stars. They're waving around and walking around. They're detrital feeders, but I'm not sure what these are or what they're doing.
And about how far under the ocean is this?
Sorry, how far under the ocean is this?
Yeah.
I don't remember what site this is, but our sites ranged between, I'll pull this up real quick. Our sites ranged between 650 meters, or actually 400 meters in Baltimore Canyon up in the Northeast to 3,300 meters or more.
Wow.
This says Florida Canyon. This is actually Florida Escarpment. Mm-hmm.
And then the last question in the chat is like, is there a time limit for being down that deep? Like, does it change with depth?
Does it change with depth? We're more or less limited by the hours of daylight because we don't want to do night dives. You could theoretically we could stay down there for a couple of days. For example, if there was an emergency, we could, and there's air supply, and we'd probably be most strapped for food. I think there's water too.
Wait, Alvin is equipped to stay underwater in deep sea for days.
Yes, it can. Yeah, it has all the air in there. I think somewhere in here I've got a picture of the interior of Alvin. Here we are. Well, you can't see it, but there's supposed to be underneath there are a ton of air tanks, air bottles. Yep.
Wow.
So you can see the gas masks there. That's how we can, we just protect ourselves from any smoke that occurs, not that it does. Some other pictures. Yeah, Alvin's equipped to stay underwater for a very long time. We try to get out into the water as early as we can, and we come back as late as we can. The limitations that are imparted to us are primarily weather, and like I said, the hours of daylight.
Wow, this is awesome. Yep. Well, it's 8 o'clock, so we should wrap up but this has been awesome. I've learned a lot. I feel like I went down to the deep sea for a brief moment there.
I'm glad to share that with you.
Yeah, no, I'm excited. But thanks everybody for tuning in. These happen every Thursday night at 7 o'clock. I am usually not here, so next week Chris will be back with you. But thanks again, Ian. This was awesome. Yeah, thanks for sharing your stuff with us.
It was a pleasure. Thanks.
All right, bye everybody.